Welcome everyone, and I would like to welcome Fengchao Yu and Rowan Beck. They are presenting on FragPipe and how it enables one-stop proteomics data analysis. Today's webinar series will focus on proteomics, and we will have Dr. Fengchao Yu, a researcher at the University of Michigan, introducing us on FragPipe as an application, and as he's the leading developer and maintainer of FragPipe, MSFragger, and IonQuant. He's currently a research investigator at Alexei Nevishky's lab at the University of Michigan. Rowan Beck, she's a community engagement manager at Seven Bridges, and she will show us how the FragPipe workflow works on the Cancer Genomics Cloud. So not only we will have an introduction on the actual software, but also how we could run it on our platform. So we will do a 30-minute presentation from Dr. Fengchao Yu, and then we will move on to Rowan, where she will show us a demo on the platform, and we will have some time for questions. And in the end, you can put your questions in the chat, or by the time when we're done with the presentation, if you'd like, you can unmute yourself and ask questions. It's supposed to be a discussion, so hope you enjoy it, and we'll let Dr. Fengchao Yu take it away. Thank you so much.
Thank you for your nice introduction, and thank you all for the invitations. It's my pleasure to present our work in the Seven Bridges and the CGCs. So in this webinar, I would like to talk about the FragPipe, which is a proteomics data analysis suite from our lab. Before I really go to the FragPipe, let me give a very brief background about the data acquisition and the analysis in the bottom-up proteomics. So in bottom-up proteomics, proteins are first digested into peptides, and then those peptides are fed into the instrument to generate mass spectrometry data. So normally there are three types of the data acquisition protocols: data-dependent acquisition, targeted, and data-independent acquisitions. So those three types of the data acquisition protocol will produce different types of mass spectrometry data. Thus, several tools were developed to analyze those kind of data separately. And then this figure was in a reviewed paper published in 2016. Then in 2017, the first public version of the FragPipe was released. And then at that time, it is really not called FragPipe, it's called MSFragger GUI, because it's only for running MSFragger and profits and general report. So the initial purpose of developing this user interface software is to make running MSFragger easier. But since its first release in 2017, and we keep adding more modules and functionalities to FragPipe to make it easier to use and make it more versatile and comprehensive. And then up to this week, there are already 18,000 downloads from all the versions of FragPipe. Then after this very brief history of the FragPipe, let's take a look at what the FragPipe really looks like. This is basically a screenshot of the FragPipe user interface. And from the latest version, as you can see, there are several tabs for the different tasks of the data analysis. They are like MSFragger for the identifications, there are validation for the FDI estimation, and then PTM analysis, quantifications, spectral library buildings, and DIA analysis. In the following slides, I will highlight several of the tools. First of all is the MSFragger. MSFragger is like the core part in the FragPipe. It is for the database searching for peptide identifications. And MSFragger can perform three types of the search: closed search, open search, and mass offset search. In closed search, MSFragger basically searches peptides within very narrow mass tolerance. And then this is the most conventional way of the database searching, and almost all the peptide identification tools have the ability to do that. And MSFragger can also perform the open search. In open search, MSFragger search peptides in a wide mass range, and those big mass differences can be interpreted as unknown modifications. And then we also support the mass offset search. Mass offset search is like something in between closed search and open search. So it searches a few mass offset notches. And then in these notches, MSFragger will match the fragment peaks, and then the mass offset will be interpreted as unmodified peptide if it's 0 and a modified peptide if it's non-zero. For example, if it search a mass notch at the mass equal to 80 Daltons, which assumes that this 80 Dalton probably corresponding to the phosphorylations. So mass offset search can search much more variable modifications compared to the closed search, and it also has a higher sensitivity and specificity compared to the open search because its search space is much smaller. Here's an example of the MSFragger closed search. We demonstrated to use 4 HeLa replicates from the DDA passive data, and the peaks and the max count were used as benchmarks. So this hatching bars means the search was from the semi-enzymatic digestions, and the non-hatching means it's from the enzymatic search. So as we can see, MSFragger can find more peptides and proteins compared to its competitor. We also perform runtime comparisons, and it also shows that MSFragger is much faster than peaks on the max count. So since the DDA passive data is much bigger than the Orbitrap data, the loading time actually take a big percentage compared to the total run times. So after the first time MSFragger loaded data, it will write the MS2 spectrum to a binary format. When this binary format is available, the long loading time will be largely reduced, and the total runtime will also be largely reduced too. And recently we introduced glycopeptide identification to the MSFragger. When MSFragger performs glyco search, it basically is a mass offset search. We give MSFragger a bigger mass list from the glycosylations, and then MSFragger search all those mass notches one by one to find the glycopeptides. During the search, it also considers the glycopeptide specific ions such as diagnostic ions, labile ions, non-labile ions, and capital Y ions. Those ions will be used to filter the spectrum and then be used in calculating the matching scores. We also recently developed a glycan assignment module in PTM Shepherd. PTM Shepherd is another tool from our lab. It's initially for the PTM analysis and summarizations, and then it has a glycan assignment module that can use multiple features such as oxonium ions, capital Y ions, isotopic error, and mass to infer the glycan compositions. And then it also calculates the glycan scores. This glycan score can be used to estimate the glycan FDR to measure the confidence level of the inferred glycan compositions. After talking about the MSFragger, let me introduce you a new tool from our lab called MS Booster. MS Booster is to increase the peptide ID after MSFragger search. It basically takes the output from MSFragger and then generate the MS2 spectrum and the retention time using the deep learning predictions. Those predicted MS2 spectrum and retention time can be used to calculate additional scores such as the spectral similarity and the retention time differences. Those additional scores, along with the original score from MSFragger, can be used to build a better model to increase the sensitivity. Our experiment shows that MS Booster can increase the peptide ID by 5 to 30% depending on different types of the data types. Here's a typical example of analyzing HRA peptide using MS Booster. This Venn diagram shows the result without and with MS Booster. There's a fairly large overlap between those two results, and then we analyze the motif of those additional 26% peptides. And the motif shows that it is very much similar to the ones from the shared peptides. Then we think that those additional found peptides are mostly to be true. After talking about the peptide identifications, let me move on to the quantifications. We developed a tool called IronQuant to perform the label-free and isotopic labeling quantifications. This is a diagram shows how ion quant extract the features and then perform the match between runs. Basically, with input data from Orbitrap or from DDA passive, it will build a 3D or 4D index for all those peaks, and then with this index, ion quant can find the features, trace and find the features very quickly, and then report the intensity around with other information like the rotation time boundaries, ion mobility boundaries, and so on. After finding all the features for all the runs, ion quant can perform the match between runs. Match between runs basically is for transferring the peptide from one run to the other. By transferring the peptide across all the runs, it can fill in all those missing values and to make the data more complete. And giving a run that is going to accept the transferred peptide, ion quant will calculate the correlations against all the other runs, and then it picks the highly correlated runs as a donor run. For each donor run, it will transfer the peptides to the accepted run based on their retention time, mass to charge ratio, and ion mobility if the data is from DDA passive. And then after it transfer a real peptide to it, it also transfer decoy ones with a very big M/Z shift. Because it has a very big M/Z shift, which means that the transfer decoy peptides are definitely to be false. And these decoy peptides will be used later on to build models to estimate the FDR for the match between runs. So with the decoy and target ions, there are actually 4 types of ions. So the plus one is the transfer target peptide, and that peptide do not have any MS2 match result in the accepted run. This type of the ion is basically what we want finally. And then there is also -1. -1 is like the decoy ion and don't have any MS2 match result. And then there are also +2 and -2. +2 basically is the target ion or target peptide, and this peptide also has a already have a MS2 peptide match in the accepted run. Since we already have the MS2 match result, and we have a very high confidence to believe that this type of the target peptide is true. And then there's another -2 peptide. -2 peptide is a transfer decoy peptide, but it has a MS2 match in the accepted run. Since it has a MS2 match in the accepted run, but we believe we know that the transferred peptide has a very big M/Z shift. So this type of the ion is very much likely to be false. So with these 2 high confident positive and negative ions, we can build a ALDA model to do the scorings. And then this model can be used to score the +1 and -1, and then build a mixture model for FDR estimations. So please note that this FDR estimation is not for the peptide MS2 match, it's for the match between runs. Then let me show you why do we need the FDR control for the match between runs. Here's a single cell data set from the Orbitrap fusion rumors. There are five experiments with 0 cells, 1 cell, 3 cells, 10 cells, and 50 cells. Because it's a single cell experiment, the IDs from the MS2 match are very limited because of the low abundance. Then people try to transfer the peptide from the back cell runs, like the experiment of the 50 cells, back to the single cell runs to boost the IDs. Then here's examples with the max quant and ion quant without the match between runs. Neither max quant or ion quant found many proteins from the 0 cell runs because these experiments are have 0 cells. All those tiny amount of the proteins are very likely to be from the contaminants. But with the match between run enabled, max quant found a lot more proteins. As I said, this is 0 cell runs, so those proteins are very much likely to be false. But with FDR controlled ion quant didn't find too many false proteins from 0 cell runs. This figure demonstrates that we do need the FDR control for the match between runs when the sample are sparse, like the single cell data or the low input data. Then we perform another comparison is against the max quant to demonstrate its precision. We basically perform an apple-to-apple comparison against max quant with and without the match between rounds. And then there's also another very important parameter called a minimum peptide. It basically requires the minimum number of peptide for the quantified peptide, for the quantified protein. So with minimum 1 peptide, there are more proteins quantified, but with worse CV. But by increasing the minimum peptides, the CV is better, but the number of the proteins are fewer. Then we have the similar parameter for the iron quant, and then the apple-to-apple comparison shows that iron quant can always quantify more proteins and with better CV compared to the max quant. So this is kind of a benchmarking of its precision. Then we perform another experiment to evaluate its accuracy. We use a mixture of the proteins from 3 species: human, yeast, and E. coli. And there are 2 experiments here, A and B. The ratio of the human proteins in between A and B is 1:1, and the ratio of the yeast is 2:1, and the ratio of the E. coli is 1:4. So basically, we know the ground truth ratio of those proteins. Then we can see how good those tools can recover those ratios. Again, max quant is used as a benchmark. And then, as we can see from this figure, iron quant found more proteins with much fewer outliers than the max quant. So basically, those 3 experiments demonstrated that we have a better performance than the max quant, which is widely used tools in the community. Then after talking about the label-free quantifications, let me show you another part of the iron quant, which is isotopic labeled quantifications. This is a paper published with our collaborators in UCLA. Basically, they are light and heavy labeled isotopic probe. They labeled the samples with no ratios and then mix them together and then perform an analysis using FragPipe and Skyline. And the data also have FAMEs. Some of the data also have FAMEs and some don't. And then since the mixture ratio is known, so we can evaluate how good those 2 tools are. And then these 2 figures shows that those figure panel B and panel C shows that FragPipe, this FP, which is FragPipe, performs slightly better than Skyline, which is indicated by SK. And then after talking about the DDA data analysis and quantifications, let me introduce another new functionality in the FragPipe, which is DIA data analysis. Recently, we added the DIA data support for the MSFragger because initially for MSFragger is for the DDA data database search, and then we make it also be able to search the DIA data directly without any feature extraction or retention time prediction or spectral library prediction. There's no need for all of them. So if users have the DDA data only, they can search the DDA data with MSFragger and then analyzed by the MS booster calculator and then filter the peptides with 1% peptide protein FDR and then build a spectral library. And this spectral library can be used later on as a template to quantify the data from the DIA. And this is if user only have DIA data, MSFragger can also search it and then build a spectral library and for the quantifications. The interesting part is if user have both DDA and DIA data, then both of the data can be searched by the MSFragger and then all the way down to the spectral library. This library can be used to quantify the DIA data. And then here is like preliminary result of the DIA data analysis with MSFragger and FragPipe. We use SpectralNut and DIAN standalone as benchmarks. There are 4 experiments in this data set. And the lymph node experiment only has human peptides, and another 3 experiments has a mixture of the human and the E. coli peptides. So since all of the 4 experiments have the human and E. coli peptides, then the spectral library will have the mixture of the human and E. coli. And then we use this spectral library to quantify the peptides from the DI data. We are supposed to only find human peptide from this lymph node experiment. So if there are any E. coli peptide from this lymph node experiment, those peptides are supposed to be false. So then these purple ones, which is indicated the E. coli precursor from lymph node experiment, can be used to indicate the false positive rate of those tools. And then the rest of the three colors are from the mixture of the lymph node and the other three mixture of the human and E. coli. So this can be used to evaluate the sensitivity of our tools. So spectral not direct DIA and is used, and then DIA is also used. Basically DIA will predict the spectral library from the FAST files and then use that library to quantify the precursors from DI data. And then FragPipe coupled with MSFragger are also used to evaluate performance. This FPMS1 means that only DIA data are provided in this analysis. This FPMS2 indicated that both DI and DDA data are used to build a spectral library, and then this spectral library is used to quantify the precursors from DIA. And we can see here, we can control the false positives as good as DIA, but we have a higher sensitivity when quantify the true precursors for human and E. coli. An interesting part is that with both DI and DDA, we can quantify even more compared to DIA and spectral not. And the other experiment also shows that by combining DI and DDA data together to build a spectral library, will always result in more quantified peptides and proteins from the DI data. And this is a work under supervisions. So above slides are just a brief highlights of part of the tools in FragPipe. Basically, FragPipe right now have the full capability to analyze all kinds of the data, from DDA to DIA, from the conventional search to the open search, from the proteome peptide to the glycopeptide. We also have the deep learning-based rescoring to boost sensitivities. We of course have the FDR estimations. We also have the ability to perform label-free, isotopic labeled, and isobaric labeled. And due to the limited time, I didn't talk too much about the isobaric labeled, but it really performed much better than the other two according to our benchmarks. And then we also have many built-in workflows for user to easier to load and drop in to analyze the data very quickly. And then people may ask, what about the command line interface? Because as I showed in the previous slides, FragPipe is a GUI interface-based software. And then as more and more users try to use FragPipe, they have the requirement that they want to run FragPipe in the HPC clusters or put it in web servers like what CGC is doing, or processing many jobs in batches. Then we are listening all the feedbacks, and we try our best to make the users happy. So we added this headless mode support for the FragPipe. The headless mode actually uses exactly the same code base of the FragPipe GUI mode, which means that the result will be identical when you run the headless and the GUI, and then the exact same command and the data will be processed. And then with this headless mode, a lot of things can be done. A lot of interesting things can be done. For example, people can put them to their clusters to let other users to use, and processing many jobs in batches without go back and click it. And the last but not the least, I would like to thank all the lab mates from the university group, especially acknowledged thanks for the Alex University, which is the PI of the group, and the Gods and Dimitri and all other people. I also would like to thank our collaborators, including George, which is the developer of EasyPQP. EasyPQP is a tool in FragPipe. It is for the spectral library generations. Also, Lucas Carr for the calculator. It is also in the FragPipe, and then David Steinberg, which is the developer of PTM Profit. and Bedim for the DIA. So all those 4 tools are already in the FragPipe to facilitate the analysis. We also have the other collaborators from UCLA and Germany and Germany and the PNELs. And also we would like to thank the funding agency, including NIH and NCI. Thank you very much.
Thank you so much. So we're going to switch now. After this wonderful presentation about FragPipe, we're going to have Rowan. If you could stop sharing. Sure. Rowan can share her screen. I think she's going to do a very brief presentation on what FragPipe looks like on the CGC, and then she'll do a quick demo.
Hi, everyone. Give me just a sec, and I will share my screen. Okay, let's start the slideshow. Okay, so thanks for attending our webinar. That was a great presentation. So I'll be doing a live demo of FragPipe in just a few minutes, but first I wanted to go over some background information for everyone. I wanted to start by thanking the various groups that have worked together to make this a possibility. That includes the 7 Bridges team, the Nesvitsky lab at the University of Michigan, the folks over at PDC, and of course the CGC users and collaborators. Special thanks to the NCI, which allows 7 Bridges to do wonderful things as part of the Cancer Research Data Commons. And with that, I will move on to the agenda. So first, I'll present to you some context for the data we'll be using, and then go over the platform-specific components we'll use for the analysis. Finally, I'll do a live demo and show you just how easy it is to run a project using CGC. As always, feel free to ask questions throughout the presentation. Myself or Zelia will try our best to answer them. You can type them out in the chat, and we will be happy to reach out to you. Okay, so let's begin by talking about kidney cancer. It's one of the most common cancers in both men and women, and there are an estimated 79,000 new cases in the US with almost 14,000 deaths, and that's in 2022 alone. You may be asking yourself, how can I analyze the spectral data associated with clear cell renal cell carcinoma? Just like this guy over here. So maybe as a researcher, you want to compare it to certain gene signatures. Maybe you're a clinician looking for biomarkers. Either way, you want the spectral data. Well, you're in luck. The proteome and phosphoproteome data is available in the Protein Data Commons, or the PDC for short. So in addition, you can find peptide spectrum analyses and protein summary reports. So the PDC is part of the Cancer Research Data Commons, the CRDC infrastructure, and their goal is to democratize access to cancer data. So by visiting the website, you can obtain any kind of protein data that you're looking for. So using the CGC, you can easily import this data from the PDC, which I'll show you in just a moment. And you can process it using the many different tools that we offer, including the one you just heard about in the first half of the webinar, FragPipe. You simply need to find and select the data you're interested in, and then download the manifest from there. So that is a CSV file. Once you're on the CGC platform, you can select Add Files and then select that CSV file. So I'll show you this in just a moment, but it really is that simple. Okay, so let's back up just a second and talk about proteomics as a whole. So in a traditional proteomics workflow, you basically have a peptide and perform a mass spec analysis on it. From there, computationally, you compare the resulting spectrum with that of a theoretical one to get a list of peptide matches. You perform further computational assessments to find and assign a peptide as the best match to your spectrum. So for the quantification step, that's a little bit more complicated as there are many different workflows. You can label proteins to infer the identity and obtain a relative quantification. You can perform a spectral counting, again, to obtain a relative quantification, or you can analyze and perform a quantification of MS data and then only identify those spectra showing differential expression. So again, many different ways to do this. Earlier in the webinar, you heard Dr. Yu speak about his lab's workflow, FragPipe. So this is a pipeline developed by the Nesvitsky lab at the University of Michigan. It contains 3 parts. MSFragger, which identifies peptides based on the spectral signatures. Philosopher, which then goes on to process the MSFragger results and quantify the peptides. And then lastly, we have TMT Integrator, which analyzes the abundances and generates reports. All of these tools and more can be found in our public apps gallery just by searching FragPipe or any protein-related keywords. I think if you type protein, it comes up. Today, we'll be using 4 workflows in our FragPipe protein analysis, as you can see right here. As always, if you get stuck or don't know where to begin, or if you just want to chat, please email me or Zelia, and we will be happy to get back to you. We also have office hours twice a week, so please drop in and we are more than happy to assist you. Okay, so I am going to switch over from my slide deck onto the CGC platform. Give me just a moment to do that. Okay, so once you are logged into CGC, this is what it will look like. You'll be taken to your homepage. So this is the main homepage. List of projects is on the left, and then analyses and tasks are on the right. Mine might look a little bit cluttered. Yours doesn't have to look the same, but you can scroll down and see an overview of public apps. There's even a useful getting started guide here. So there's a quick start. You can create a project, invite collaborators. So what I'm going to do is create a new project. So you can select projects from the top and then create a project on the bottom. We can name it anything we want. Let's name it FragPipe demo. And then the date 8/24, and then select the billing group. So I'm going to use my pilot funds. Keep in mind, anyone who signs up for a CGC account will be given $300 worth of credits. So that's all you have to do is sign up, and you get credits to play around and do any analysis that you're interested in. You can leave everything else pretty much the same, and you'll create your project. Okay, so from here, you'll be taken to a new dashboard page. On the left, you'll see a description. You can edit this yourself with Markdown. On the right, you'll see a list of project members. You can easily invite members by searching for them or using their email address. Here, I'm going to add Zelia. And I'm going to give her write, copy, and execute access, but not make her an admin. And I will invite her to my project. And she is now added to the project. On the bottom here, you'll see various tasks that have been run. Since we just created this project, obviously there are no tasks that have run, but we can fix that. Over on the top here, you'll see a Files tab. This allows you to populate. There are no files right now in our project, but let's say you wanted to obtain some files. You could go over to the PDC. So this is the homepage here for the proteomic data commons. And again, we're interested in clear cell renal cell carcinoma, which happens to be right over here. You can click on that, you'll be taken to a new page. Okay, you can scroll down and select the type of studies that you're interested in. I'm looking for, let's see, the Files tab right here, and then I want open studies. So these have no restrictions to them. I'm gonna just select like 2 of these. And then what you'd like to do is click on export file manifest. So you want that as a CSV. Once you do that, it should download here at the bottom. Go back to your Files tab, and then what you want to do is click Add Files. So add files and then import from a manifest file. So what I'm going to do is click and drag. I'm going to even label it with a tag. Let's say PDC. And then let's do another tag that says manifest so that we're all aware of where these came from. Click import and then give it just a moment. So you'll see the actual CSV file here has been uploaded. I'm going to click back to my dashboard and then files. And now you'll see that the files from the PDC have populated in our project. From here, you can go to the apps, and then you can add an app from our public apps gallery. So again, we can search for FragPipe, and you'll see several different apps. We're going to copy these all to our project. I would like to copy this one. You can select as many as you'd like. And then go back to your apps tab, and now you'll see these 4 that you've just copied over. Lastly is the tasks tab. And here we have no active tasks running. So you can fix that by going into your apps, selecting the very first of the 4 that we'll be using. So this is convert, identify, peptide profit, and click run. And then from here, you can select your raw files and the database files. Let's say you just want to do something very simple for today. We actually have a public project here with FragPipe. So what you could do is click on the public projects over the top here, and then scroll down, click FragPipe Proteomics. From here, click the little eye icon, and you can actually copy this entire project over and use that as kind of a template. So I'm going to make a copy of this project. And now instead of the demo project we were working in, we have this new copy of FragPipe proteomics. You can see the files that they had used. There are apps, just like we added. And then most importantly, tasks. So this is one of my favorite things about the CGC is if you don't really know where to begin, you can use another project as kind of a template. You can click on any of the tasks that have been run, and then you can either edit and rerun the exact task, or you can go back to the apps and run it yourself kind of from scratch. So I'm going to run the very first one from here. Okay, we are going to need a database file. Once you click add file, it will come up. I'll show you that again in case I did it too fast. So you'll click either select files or change selection. Because it's a database file, it has to be .fas, and the system automatically will only give you that option. So it knows that that's what we need to use. For the raw files, we have a large number here, more than just the two we copied over from the PDC. So again, you'll need raw files here. So the system automatically filters everything and only shows you the raw files. You can select all, save selection, and then pretty much you keep everything default. What we're going to do actually is batch these files. So what that means is running them simultaneously. And we're going to batch by the metadata of the files. So if you'll scroll down, you can click on plex or dataset name. So that divides all the files by experiment type, and it just runs faster. So again, keep everything else pretty much default. You're going to scroll down, and there is one thing that you will need to, if I can find it here, there's an agreement that you need to accept. Oh, but because it's a public project, I think it's already been accepted. So that makes sense. If you're doing this from scratch and adding it, there's just one drop-down menu here that you have to click yes. So what you'll want to do is click run, and it'll tell you you're about to run 4 tasks because this is divided into 4. And you can click on run. While that's running, we can actually view already completed tasks. So here is the one that was completed. Okay, and you'll see what it looks like once it's completed. You'll just have some output files. So on the left here, you'll see the input files we've selected, and on the right are the output files. These are .tar files. And then you'll go on to the next step and do the same thing. So once again, we have input files, which are the outputs from the previous step, and then we'll end up with an XML file as an output, and then so forth. So go on to the third step. And once again, we are batching these reports. You can pretty much run these in the same order, and again, use the tasks as templates. And then you get TSV files as the outputs, and these go in to the input of the final step, which you'll see as the TMT integrator and QC run. So again, you'll include the database file as well as the .tsv files. And this is pretty much the final step. So you'll get output settings here, including a gene report, and this is an HTML file. So you can click this and view it right in the web browser. It gives you protein intensity, variance, and distribution. So here are box plots that are generated, as well as just a different way of viewing this. There's clustering that it provides a heat map on the samples. And lastly, what you can do is go into Data Studio and create an analysis. You can select either Jupyter Lab Notebooks or RStudio, give it a name, and you can analyze the data yourself if you want to go and look further into it. So I already have one of these running actually right here, and I have an RStudio. And from here, we have an R Markdown code that just kind of reproduces the last HTML report. So here I can run several of the chunks and generate plots. So one last one. And here we have the box plots once again. So if anyone has any questions or would like me to repeat any of this, feel free to ask. I believe that is all for the demo for right now.
We have a question on the chat. Could you clarify if that's for Rowan or for Dr. Yu? It says, this is very similar to the previous implementation of FragPipe. What is the difference? For Rowan.
Honestly, I'm not an expert on the FragPipe, so Dr. Yu could probably speak up about the latest version. I know there is a new version that we're hoping to get on the CGC, but I don't know the differences between this and the previous one.
Well, I believe when I see the screenshot, we can screenshot from CGC. I believe it's from the FragPipe 15. Right now we have the FragPipe 18, which basically have more functionalities compared to the other versions, as I presented like at the beginning. But yeah.
Oh, so they're mentioning the CGC workflow that was added a couple months ago. Could you clarify? You can unmute if you'd like. We can just talk. Sorry. You do? Yeah, so I'm not sure which CGC workflow that you were mentioning. Maybe we can chat a little bit offline after this so we can properly answer your question. I'm unclear what you're referring to. Of course. Does anybody have any questions? Rowan, do you have any questions for Dr. Yu, vice versa, and somebody from the audience?
Sort of a question I was going to email later, but basically we're really excited to get the latest version on the CGC. So I just wanted to say that I will be working with you later on to try and update this so that it's the most recent up-to-date version.
Sure.
And I'm looking forward to doing that and, you know, testing it out, noticing differences.
Sure.
So I guess I also do have a question for Dr. Yu. I believe you were helping with the implementation of FragPipe. Could you just share a little bit about your research in your lab and other things that you're working on that might be interesting to the audience?
You mean, so, okay, so let me talk about myself first. So what I present in the first 30 minutes is basically what I'm doing in the lab, including adding the new functionality to MSFraggers and working with Dan Pelavsky for the Glycopeptide identifications, and also the label-free and isotopic label things is also part of my research project in IonQuant. And then DIA thing is also, that is a paper under submissions, and is also my major research topic in the past year. Yeah. So for the other guys in our lab, as you can see, most of our guys contributed to the FragPipe, including the team integrators and a philosopher from Philippines and other things. And then there's another big project, which is CPTech, which is not very much directly related to FragPipe, but they are using FragPipe for it. So it's basically the situation in our lab, I guess. But of course, Alex is a better person than me to answer this kind of question.
No, that was great.
Yeah.
No, we are really interested in utilizing the PDC and like CPTech data sets, and the FragPipe pipeline is one of the most requested ones. So just interesting that.
Yeah. A bigger update in the latest FragPipe, which might related to CGC, is that we added a command line interface for it. Before there's a GUI things. Right now we have command line, so that it would be much for for you to using the latest FragPipe, because they just replace the older one with a new one, and then using one command line for it. And there are already several groups that is trying to adopt it in their own online server thing. And I believe this might be the most the biggest update that is interesting to you. Yeah. And other things as I present, I believe Roman, is that a Roman? Yeah, Roman present about the TMT data analysis. I believe that's what the CPTech is about, because CPTech is all about TMT data. But we only can do the label-free and DIA data. If CGC has interest, you feel free to reach out and we can show you how to do that. Yeah.
Yes, absolutely. We want to support as many users as possible. And so if that's where the community is going, and that's the kind of data that people are analyzing, we absolutely would love to learn more and support it in our platform. So Rowan and I will absolutely reach out to you so we can.
Sure, sounds good. I actually have a question regarding the CGC things. So I have a very practical questions. Do we need to pay if we run on CGC?
Well, yes and no, I guess is the question. So when you open your account, you have $300 of pilot credits. Usually those pilot credits are enough to run tests, and you can run a full RNA-seq experiment with 20 samples without ever using your entire pilot credits. If you want to do something a little more complex, then yes, you would need to pay, but it's a pay by usage. So for example, the more you use it, the more you pay. You can also apply for a collaborative award. So basically in these collaborative approaches, we work together to either develop a tool, or it's a more complex project that we can work on it. You would have support from our bioinformatics team. We would have check-ins. It's sort of like a grant, but more like an award. So we can have up to $10,000 to help set up that initial product. A lot of our collaborators use that as to generate preliminary data that they can then put into their NIH grants, and then can be funded through a more regular funding mechanism. So yes, you do have to pay, but
that sounds good. So if I want to analyze CPTech data, for example, are the data already in the cloud, or I have to upload it by myself?
The data is already in the cloud. So you could, Rowan, if you could switch your tab to the Proteoma Data Commons that you have open, you would go on to PDC and you would look for the CPTech data you're interested in. You would export the manifests that you have on the bottom right. And then you would upload the manifest in your project using import manifest, and it will automatically show up what's in the PDC and proteomics data commons will be on your project. It's already in the cloud.
So rather than, because when I try to analyze some CPTech data, I always have to download it to my own computers and write. So rather than do that, I can just do it online. That's
Again, just download the CSV, the manifest right here, and then you can upload that easily into your project. I only did it with 2 samples, so it took just 10 seconds, but it might take a little bit longer, but it's really very, very quick.
Yes.
Yeah, sure. It's very fantastic. And what other than CPTech, are there any other types of, like the Pride, the Massive? Are they still also online already in the cloud or not?
I would have to look. What data did you ask?
Like the data in the Pride or data in the Massive, like protein exchange.
I am not sure. I would have to look it up. So, but if you want to see, so we have data from the Genomics Data Commons, from the Integrated Canine Data Commons. We have the Cancer Data Service, TCGA, Target. We have a lot of different data. So I'm not sure where that data that you're mentioning would fit. But if you send me the name, I can look it up.
Sounds good. Yeah. Let's talk after the presentation. Yeah.
Yes, absolutely.
Very interesting. Yeah. Sounds good. Yeah.
Yes. So if you're absolutely interested in applying for a collaborative project, we can just send you an application and we can talk about it. And fairly simple process. Yeah. Do we have any more questions?
No. Yeah, good. Is there any questions from audience?
Yeah, I was trying to hope for the audience to say something. There's a question from the audience. If I have my own data to upload it in the project, does it have to be CSV? Data frame has to be formatted in a particular way. We usually receive Excel files from a proteomics core. No, you can absolutely upload your own data to the platform. Rowan, would you mind going to your account and show how to upload on your own data really quick? We're just going to do an impromptu demo here.
Yeah,
So
sure.
you can just go to the
Files.
and
then add files. So there are lots of different ways to do it. You can browse our own publicly hosted data. You can look for other public files. You can copy data over from a previous project that you had worked on. You can upload it directly from your computer. You can upload it from a computer, like a high computing cluster. Sorry, inarticulate today. Importing from a manifest file is right here. So that's kind of how I did it earlier, was just with the CSV file. If you do have it in Excel format already, you can save as .csv and try it out that way. It might make it easier, but there are really multiple ways.
So if you have your own data already, you can just go add files through your computer and you'll literally just drag and drop the file. Depending on which workflow you're using, the data set input would be different, right? For this case, the CSV file would have the manifest format, it's for the manifest, but the data that you're going to actually be using is the raw files from your mass spectrometry, right? So it will really depend on the workflow and the data that you need, but you can easily upload your own data.
There's a way to do it always. So you can always, I forgot to mention, but click on this little question mark on any of the pages on the CGC as well. And that'll take you right to our support team. And they're very, very quick. So you can just type in any issue you're having or any question, and they can respond to you like very quickly.
And like Rowan said, we have office hours on Tuesdays and Thursdays, 10 a.m. and 2 p.m. respectively. So we'll be there to answer questions. Even if you just started and you just want to understand how some of these things work, we'll be there to help.
It's very relaxed. So come on in, just, even if you don't know where to start, you just have, you're curious about something, we're happy to walk you through it.
All right. So if we don't have any more questions, I guess we can end a little bit early. Before we end, I would like to also make a quick announcement for next month's webinar series. It'll be led by Dr. Min Zhang from Purdue University, where she's going to be sharing her experience on using the CGC as a training tool. We ran a class with her earlier this spring that was very successful, and so she's going to show us how that went. And so that'll be at the end of September. And I hope to see you all there. And if you have any questions, you can feel free to email us or join us on office hours. We'll be here to help everyone. Thank you again for coming to the CGC Webinar Series. Thank you, Dr. Yu, for your wonderful presentation. And thank you, Rowan, for your demo.
Thank you.
Thanks, everyone. See you next month. Bye-bye.